What's up, everybody? So in this video, we're going to be talking about the immune system. So the way I see it is in C3.2, there's a whole bunch of stuff. And we're going to break it down into 4 videos, right? So this one is going to be a big video. It's going to be, most of the stuff will be in this video. So we call this video Defense Against Infectious Disease, because we're going to talk about pretty much just that. And there's a lot of things falling under that category. And then in the next video, we're going to be talking about the ABO and Rh blood groups. After that, we'll talk about zoonotic diseases, which is, both of these are short videos. Honestly, all 3 of these are fairly short videos. It's just, I didn't want to make this video too long by talking about all of this in one video. So in this video, we're talking about all of these things, defense against infectious disease, and then the subsequent 3 videos will be covering these 3 things. So that's the deal. So let's just get started. So what is the immune system? So when you think about your body, right? Your body is very, very, very complex, right? You've got a heart, you've got lungs, you've got kidneys, you've got intestines, you've got so much stuff, right? You've got so many systems. A system is pretty much just a bunch of organs working together to accomplish a task. So for example, one system can be your cardiovascular system. That system includes your heart and all your vessels, and it's responsible for moving your blood around your entire body so your cells can survive. Another system can be your respiratory system, which includes your lungs, right? And that one is responsible for getting air in and out of your body. So there are many systems, and this system, the immune system, is going to be the one that's responsible for protecting your body from infections, right? Very, very, very important. Without this system, you'd be dead any minute, right? Because there's always dangerous things around you that's very small that can infect you. So that's what your immune system is. So all these things that can infect you, we give them a name. We call them pathogens. Just like this bad guy, they're bad guys, right? These pathogens. So the definition of a pathogen is any disease-causing organism. So what can that include? We can divide pathogens into 2 categories. One being non-living pathogens, and the other being living pathogens. Non-living pathogens, guess what that would be? If you're HL, or even if you're SL, you should know this. It's a virus. A virus is a non-living pathogen. A virus. What else? What else do we got? So living includes more things, such as bacteria. Remember, bacteria, is a living thing. It's a prokaryotic cell. It's a prokaryotic organisms, right? Humans are eukaryotic. A bacteria would be prokaryotic. We've also got fungi, and we've also got parasites. So these ones in blue are all examples of pathogens, right? Virus being non-living, and these 3 being living. Bacteria can be good and bad. So you can have bacteria that is helpful to your body, and you can have bacteria that is bad for your body. So when a bacteria is bad for your body, in that case, it is a pathogen. In other cases where it's helpful for you, like some bacteria actually help protect you and helps digest your food, right? These are good ones. So in this case, we're talking about pathogenic bacteria, bad ones. Fungi, I don't know, I have this, I often in the past had this problem where my feet, in between my toes, I would get athlete's foot, which is just like this infection that makes you in between your toes get infected with a fungi, and that causes like soreness and problems, and you have to like obviously take medication to make it go away and not scratch. It's super, super itchy. I don't know if you've had this before. That's caused by fungi, which is also a kind of pathogen. And then parasites, right? Some parasites can be huge, can be like worms, right? And some can be small. These are like, for example, like 30 centimeters long. Like they can be inside your intestines, and it causes severe pain and bleeding when you need to use the bathroom or whatever, and then they need to be removed, right, by a doctor. So all of these are pathogens, right? Pathogens. Just make sure you know these 4 pathogens, know the non-living ones, know the living ones. Very important. Now, you should be very experienced with this at this point, with the whole COVID thing. So what's the best way to not get infected by a pathogen? The best way. Is it medication? Is it preventative medication? Is it some ninja moves to destroy them in the air before they get to you? No. What's the best way? You should know this from COVID. Quarantine, right? Getting isolated, physically running away from this pathogen is the best way to not get infected in the first place. So make sure you know that self-isolation is important with these infectious diseases, because the only way they can infect you is by getting in contact with you and getting into your body. So if you run away like a little girl or a little very brave person running away, then you'll probably survive better than someone who's stupid and decides to encounter them, right? So no quarantine is very important. Okay, so now I want to give you this analogy, and if you understand this analogy, you're going to understand the real scenario very well. So here I have a wall, okay, a wall. On this side of this wall, we have an army, right? So this wall is protecting them from the outside, where you have other armies, you have other countries or other people, right? We're going to consider these the bad people that don't belong within the confinements of this wall, and these are the good people, including the ones on the walls that are protecting what's ever inside this wall, okay? So think about it. What is a very important thing keeping these people out of this, out of the confinements of what's going on here? This wall. This wall is the first barrier. The only way these people can get into here and start causing trouble is if they pass this wall. Your body is super similar. Let me show you. Your body has a first line of defense, and we call that first line of this defense your skin. So your skin is going to behave a lot like this wall, and these guys are like the pathogens, the bad things that can infect our body. And this right here is representing the inside of our body. So you can keep this analogy in mind, and if you forget things, you can always refer back to this analogy and understand it again. So let's look at our first line of defense, our skin. So our skin is covering the outside of our body, right? The way things can get into our body is two ways: through our skin into our bloodstream, or through other openings. So we have a mouth, we have anus, we have urethral tract, right, where you pee from. You got a nose, you got ears, right? You got all these openings. Now, your skin and your openings are made up of a different material. Like if your skin is made of a certain structure, and the inside, like if you feel the inside of your mouth, where your airways, and where you pee from, it has different structures, and we're going to look at that now because we need to know how it's made to know how it's helping our body. So if we look at the skin has two layers, dermis and epidermis. So dermis is a layer of dead cells. That's why I put these cross here. They dead. Okay, they're dead cells. So when you feel your skin, those are actually dead cells. The very top layer is dead cells. But still, they're important because they behave like this wall that I just mentioned. So if you have all these bacteria or pathogens on the outside, they first got to cross this dead wall, this layer called the epidermis. It's called epidermis because epi means on top of, and dermis is this layer. This is layer right beneath it. So epidermis is called epidermis because it's on top of the dermis. So if you look at your skin, the top layer, the most top layer is called the epidermis. Right beneath that is called the dermis. And inside the dermis, this layer, is a bunch of blood vessels. So here's a blood vessel running along, and this is another vessel that we're looking into, right? It's made up of vessels and some cells, some small cells. I didn't show them here, but there's a few cells here as well. And there's some nerves and things like that, okay? Very important. But the point is, this pathogen at the end of the day needs to cross your epidermis before it can get into your bloodstream, right? Its target is your bloodstream. Once it's in your bloodstream, it can go and travel all around your body and infect some place that it wants to infect, like your lungs or whatever, right? It can go anywhere once it's in your bloodstream. That's its target. Okay. Now, this is your skin. This is your first line of defense. The bacteria or whatnot needs to break through this first. Now, what about your openings? Your openings is different, like your inside your mouth and stuff, because bacteria can float inside your mouth and then they can get into your body, right? So your openings are a bit different. Your openings doesn't have the exact same structure, but it's a similar concept. So when you feel inside your mouth, it's very slimy, very different, right? It's different from your skin. So what's the difference in the structure in the openings of your body, like your nose, your throat, and all that, compared to your skin? The difference is this. Instead of having an epidermis, instead of being like your skin, the structure is a bit different. The most surface layer is the mucous membrane, and these cells are living, and they make something. They make this mucus. That's what we call it, the mucous membrane. This membrane of cells are alive, and they make this mucus, this slimy stuff. So when you stick your hand into your throat, that's what you're feeling, this mucus slippery layer. And it's very important because guess what it does? It traps these bacteria. So for example, if this bacteria is swimming around and gets into your throat, the first thing it's gonna want to do is try and get to your vessels, and they're gonna go and get stuck, can't go anywhere. And when they get stuck, some cells from your immune system can come and destroy them or get rid of them. Okay, so that's very important. Your mucous membrane can make this slimy mucousy layer that helps trap these bacteria. And this mucus layer also has some stuff in them, some molecules that can also attack and destroy these bacteria. And a good way I like to think of this in terms of the analogy is, remember, you've got these guys inside of your body or inside of this castle that's there to protect the castle, but you also got these guys on top of this wall that can shoot and protect things and prevent them from coming to the wall in the first place, right? These guys can shoot their arrows, shoot their guns, and kill these pathogens before they even get to the wall. So if you look at this here, in a way, that's what this mucus layer does, is it protects, it stops these bad guys, these pathogens, from even reaching the mucous membrane or the wall in the first place. That's different. The skin doesn't really have that. So that's special about the openings of our body, the slimy parts. Okay, but anyways, so the aim of these bacteria was to get in here, but first it's got to cross this mucus layer, these cells, and then they can get into there. So that's it. That's it for our first line of defense. First line of defense, very important. So no, at the end of the day, we start with this bacteria. It's going to try and get into our body, but the first thing it's got to do is pass our first line of defense. If it can't do that, it can't get into our body. Okay, so that's very important. First line of defense. So here, I want to show you something before we move on. Because I'm going to be using a lot of cells and little molecules and things like that, I have this page here. It's going to be the character profile. So because I'm not going to put the names everywhere in every diagram, I'm not going to label them because I don't want to make it too overwhelming. Like, don't have too many words on one page. Here I have this character profile. And if you ever look at a thing and you forgot what its name is, you can come back here and quickly look at this page. It should have all the names and things so that it's clear for you. So let's continue with the immune system. So we covered now what a pathogen is, right? What a pathogen is, the thing that wants to kill us. And we've covered the first line of defense, the first thing that they got to get through, our skin, okay? Or our barrier. Now let's continue with this analogy. So let's say this wall gets destroyed, okay? Let's say for some reason they managed to be absolute ninjas and avoid all these gunshots and now they're here and they break the wall. Or the wall is broken for some reason. Maybe they didn't break it. Maybe the wall got old and it broke. Or maybe they had their own weapons and they shot the wall and it broke. Okay? So what's going to happen if this wall is left open? These guys can very easily come through. They can very easily run through this wall, get in, and start causing trouble in our body, right? Or inside this castle. So it's very important that what happens? That this wall is fixed as soon as possible. So how does that relate to our body? So remember, we have this layer, right? These dead cells, this epidermis. Right? And just like this wall can break that I just talked about, our skin can break. So our epidermis can get sliced. Say someone attacks you, the side you're walking around at night where you shouldn't be, and someone brings a knife and they slice you, right? If they slice you, maybe your skin gets broken, your skin gets damaged. Now it's open, and maybe your blood vessel gets cut. So now you're bleeding, right? Now you're bleeding. Now there's this opening, a very nice opportunity for these bacteria to come in, waltz in, right, and destroy you. So that's very important. So we want to close up this hole, this damage. We want to clot our blood. We want to clot. That's what, when you cut your blood, when you cut your skin, it starts bleeding, and after a while, it clots up and it starts getting hard, that area, right? It healed. You did some blood clotting. That's important because we got to fix this hole so that no more bacteria can come in, right? It's kind of the reason why our body can naturally close this, but it's also the reason why when you get cut, you put a Band-Aid or a plaster, right? Which is kind of helping us close it while your body is trying to close it, right? Because while maybe your body is pretty slow at closing it, if you put a Band-Aid, you can close that and prevent these bacteria from coming in. So that's kind of the use of them. It's not to make you putting them on doesn't mean you're sensitive or something. It just or you're a baby. No, it's good because it prevents these bacteria from coming in. If you're gonna be all tough and not put a plaster, you're more likely to get infected and have big problems afterwards. So it's not about toughness or anything like that. It's useful to cover it up, right? It's like an artificial barrier, right? Okay, great. So let's say this is cut open. How does our body fix it? How does blood clotting happen? Very, very simple. So in your blood vessels, you got all these things. Now, so this is in the dermis, remember? The dermis is the layer beneath the epidermis that has a lot of cells and vessels, like these blood vessels. This is a nice blood vessel. This is a blood vessel. We're looking at a big one here. And in the blood vessel, we have a lot of cells. Okay, so this one right here is our red blood cell. This is the reason why our blood looks red, because our red blood cells, when they carry oxygen, they emit red light. I mean, they don't emit red light, but they appear red when they carry oxygen. So therefore, your blood, when you're bleeding, it's red because of this guy, the RBC, or the red blood cell. And again, come back to these pictures here. You can see I label all these things to make it clear. Okay. Okay, so you got your red blood cell. What else we got? We got this. This is a white blood cell. So white blood cells are not as simple as red blood cells. There's not a single cell called white blood cell. A white blood cell is a category of cells. So let me go back to this image. White blood cells, also known as leukocyte. By the way, red blood cell is called the erythrocyte because ereth means red, cyte means cell. So it's a red blood cell. Leukocyte, leuko means white. So it's a white blood cell. There are many kinds. So that's why I put this picture, because it could be any of these cells. It could be thousands of cells. And in the IB, you need to know about these three kinds of white blood cells. So the B cell, the T cell, and the macrophage, they are kinds of white blood cells. Okay, so that's why I put the picture like that, because I'm not specifying any specific one. Okay, so that's a white blood cell. Then we have more red blood cells. We got this little guy here called a platelet. Platelet, it's going to be very important in causing this clotting to happen. And we got these two molecules here. Okay, and we're gonna mention their names very shortly. Okay, so let's explain. These are proteins. These are little proteins. They're not cells, they're proteins. And this platelets is like little cells, the little pieces of cells. Okay, so let's now describe blood clotting. What happens when you get cut open like this? So your blood is flowing, you gotta imagine. So you're flowing, new cells keep coming. It's a flowing process, right? That's why when you cut yourself, you bleed, because your heart is pumping blood continuously, and so more blood is flowing out. Very important. So what happens is these little platelets, they are little surveillance guys. So they move and they can sense, or they can detect places where there is gaps. They can detect these openings. They can detect these areas where damage happened. Okay, because when damage happens, these cells here that are damaged release little molecules that this guy can smell, or they can pick it up. So he's swimming. He's like, oh, all healthy, all healthy. Oh crap, something happened here. So the platelet picks up that area. Okay, it picks up that area, and then as soon as he picks up that area, he produces a bunch of these small things, these little guys here. Okay, and they are called clotting factors, because these are little things needed to cause clotting to happen. Okay, so basically, this platelets detects this area, he releases these clotting factors. These clotting factors are useful because they do something. Let me show you. Let me move these cells out of the way. These clotting factors are gonna take. Let me move these guys. So this yellow molecule here, when it has this thing over it, this little no sign kind of means it's inactive. When this yellow molecule is inactive, we call it this. Let me show you. So when this yellow molecule is inactive, meaning this little sign is over it, we call it prothrombin. Pro meaning before, thrombin meaning this molecule. So it's the molecule before thrombin. Okay, so it's the molecule that becomes thrombin. It's called prothrombin. So the only way it can become thrombin is if these little clotting factors tell it to do so. So when we come back to this image, the platelets sense the area, release these clotting factors. These clotting factors will now go towards prothrombin, which is this one here. It will take off this sign. It'll activate it to become thrombin. Now it is thrombin. Now thrombin is gonna, guess what? It's going to go to this molecule here, which is also inactive. This inactive molecule is called fibrinogen. Let me show you. Fibrinogen. So it's going to do the same thing. It's going to go and take off that sign, and now it's going to be called fibrin. So it comes, it takes off that sign. Now it's called fibrin. Now, the important thing here. This one here, fibrinogen, is soluble. Soluble, meaning it likes to stay within the blood. It likes to stay in the middle of the blood. It's nice. It dissolves well. It dissolves well in blood. But as soon as this thrombin takes off this sign and activates it into fibrin, now it is insoluble. Now it is insoluble. When it's insoluble, it means it's not going to stay in the blood well. So it's going to go up because it can't be in the blood. It can't be in the blood. So it's suddenly going to, it's like when you try and swim with a bunch of air tubes, like with a bunch of air tubes. You're just going to float to the surface, right? Because you're going to float to the surface. So instead of being able to be in the water, now it's going to float to the surface. So it's going to float to this little hole, right? And it's going to stop there. because now it's outside of the blood, because it's insoluble. Let me put it here. It's insoluble. Whereas when it's inside the blood and has this cover on, it is soluble. Sorry, let me bring this. I'm gonna make this clear. Okay. So let me reiterate. So we got this damage, this platelet comes, detects the damage, releases these clotting factors. These clotting factors will turn prothrombin into thrombin. Thrombin will turn fibrinogen into fibrin. By turning it from fibrinogen into fibrin, it goes from being soluble to insoluble. When it's insoluble, it doesn't want to be in the blood, cannot dissolve well, and it travels to the surface. And now it's going to help clot it up. So this happens many, many times, thousands of times. And if this happens enough times, you're going to have a lot of these little guys covering up this hole. Okay, and this is our clot. This is the clot we're talking about. So now it's closed. Now blood can no longer leave. That's why you stop bleeding after a while. Now, because these are little lines, these fibrins, a lot of the times red blood cells get trapped in it, or platelets get trapped in it. So it becomes a whole clot, okay? So we call all these things, when all these platelets are there, because right, it's not only going to be one platelet, right? We have a lot of platelets floating around, and a lot of them will come to the site and stop and release these clotting factors, right? It won't just be one. So when all this collection of these platelets is called the platelet plug, okay? And the platelet plug will cause this whole cascade of reactions to form this whole fibrin thing. And this fibrin thing we call the fibrin mesh. Okay, so very important. This whole thing happens, the platelets accumulate here, which we call the platelet plug, because it's kind of plugging this hole. They cause this cascade of reactions that I just mentioned, and that leads to a fibrin. All these fibrins be coming here because they're insoluble, and we call that the fibrin mesh. Okay, so that's it for blood clotting. I want you to notice that white blood cells were not even involved in this process. Remember, you all think of our white blood cells as the key to our immune system. It fights these cells, but in fact, they're not even responsible for the very first step, which is fixing our barrier when it's broken. They're not even involved in that. They only get involved when the bacteria actually gets inside, right? Okay, so that's very good. Very, very good. I'm going to show you here a picture, a real picture, microscope picture, electron microscope picture, very small, of this fibrin mesh. Look at all these little wires. These are the fibrin molecules that we just looked at. This fibrin molecules, right, that is helping form this clot. And you can see there's a lot of other cells, like these red blood cells, white blood cells, and all that trapped inside this. Just like this whole mess I showed you here, right? So this is like a real picture. It's pretty cool. Now here I have for you a summary. So here's what it looks like beforehand when you get sliced open. Then we have the platelets releasing clotting factors. Clotting factors help convert prothrombin into thrombin. Thrombin helps to convert the soluble fibrinogen into insoluble fibrin, which causes this whole thing, right? So I hope that makes sense. That's it for the whole like clotting thing, blood clotting. So as a recap, we learned again about our pathogens. We know all these different pathogens. We know we have this first line of defense, right? And these things cannot infect us unless they pass this first line of defense. If our first line of defense gets damaged, like I just mentioned with this wall, if it gets damaged, we have a system that helps do blood clotting, and I just explained that. Okay, now what next? So we're taking this step by step. We're telling a story, right? So it's a lot of information, I know, but I hope this is making sense. I'm trying to make it as clear as possible. Okay, great, next. We got now another thing. So remember that hole. What if someone managed to pass into your body before that hole is fixed? Before you were able to do blood clotting, and something got in. What happens? This is where innate immunity happens. Now we're going to learn about our immune system, a specific part called innate immunity. It is called non-specific immunity. This is the kind of reaction that happens when something gets in and we need to respond immediately, really quick. No matter who it is, we got to kill it. This is our innate immune response, and it's also called non-specific immunity. Okay? And you'll see why it's called non-specific immunity soon. Okay, let me show you what it is. So essentially, by using this analogy, imagine someone gets in. Now, whoever this is, whatever country they're from, whatever soldier this is, it doesn't matter. Because they don't look like us, we're killing them. So just because they don't look like us, we're killing them. That's essentially the analogy version of the innate immune system. Whoever it is, whatever bacteria it is, we're going to kill it. We don't care how it looks like, how big it is, where it's from. As long as it's not one of our guys, we're killing it. That's the innate immune system. Let me show you. So here we have it again, same thing. Say we get damaged, we don't manage to clog it up in time before something gets in. So these things get in. So we have some bacteria, they get in, could be any of these. Let's just say these two got in. Any can get in. It can be this one, that one, but this one is different. It's got a orangey tail, this one's blue. So these are 3 different ones. And no matter what, let me move this one to front. So no matter who it is, we'll try one more time, if it doesn't work, then sad news. Okay, so let's say any bacteria can get in. And no matter who it is, our body's gonna fight it. Okay, first, let me take a selfie. So I want you to introduce this idea. So you have your own body cells and you've got these pathogens, right? So your body, your immune system is made in a way to notice which cells are your own cells and which cells are not your cells. So we call this idea self versus non-self. So just like that army that we looked at, these are their own people, and these are the enemy. These are the pathogens. So these people inside of this walls, they know who is their friend. Because why? Because you look at their uniform, or you recognize them. You know that's your guy. And when they see these other people, they don't recognize them. They don't look like them. They're going to kill them. It's the same way. So your body is able to recognize self versus non-self. It's able to recognize which are its own cells and which are not its cells. How does it do that? The word, if we use the analogy, that would be like uniform. You would look at the uniform. That's how you recognize it. Because they don't have the same uniform, we kill them. So in your body, in a way, the cells also have a uniform, and we call that antigens. Antigens, okay? These are little molecules that your cells look at to try and see if it's your guy, if it's your cell or not your cell. So these little pathogens, these bacteria, they have these little V things on them, okay? In reality, they can be any shape, so I just made them V for the sake of this video. But these are little molecules which your cells do not recognize. So they will see them and be like, our cells don't have that. Let's kill it, because that means it's not our cell. It's kind of like the uniform. It doesn't have the same uniform as your own cell. So all these little V things, we call them antigens. So these are molecules of these foreign and non-self entities, so these pathogens, that can trigger an immune response. So our body will see these antigens. So another way to think of antigens, as I put it here, let me find it, here in this little character profile, is antigens can be generating substances. They can be antibody generating substances. And we'll look at what exactly this means very soon. Okay, so don't get too caught up on this. Don't worry about this yet. Just know antigens are essentially uniform. Okay, wait a sec. So antigens are these little molecules like uniform, and they are on foreign things. They are on pathogens. They're not on your own cells. So your cells will recognize these antigens, and then because of realizing that they have antigens, these molecules don't look like your own cells, they're going to attack them and kill them. Okay, so remember I just said, in the innate immunity, no matter who it is, as long as they're foreign, as long as they're not your own cells, they're going to kill them. It's the same thing here. So as soon as your cells realize these antigens, they're just going to kill them. Now, which cell does that? So remember, we have white blood cells. Now, white blood cells, there's three types you need to know. The one we care about now in the innate immune response, this non-specific immune response, is the macrophage. Macro means big. Phage means eater. It's a big eater. It's just going to eat it, whatever it sees. So if we look here, what it does is it's going to go to these bacteria and just engulf them. It's going to eat them as quick as it can, okay? So that's a kind of white blood cell, the macrophage. So let me show you specifically what it does. So here's a bigger version. I'm going to show you the process of how it eats. So here's our macrophage, this whole thing, this cell, okay? And here's our virus or whatever pathogen. Let's say, in this case, we're looking at this virus. It's going to swim towards it, recognize it by its antigens, and realize, oh, this is not my guy. I got to eat it. So it's going to take it in by endocytosis. Endocytosis is the process of eating. It's basically the way of saying how a cell eats. Endocytosis. Endocytosis, endo means inside, cytosis means cell. So it's going inside the cell. It's eating it, okay? Now, the specific kind of endocytosis, so there's many ways of bringing something into a cell. This is one way, and there are many other ways. So the specific way that a macrophage does it is called phagocytosis. And basically it brings in this virus by forming a little membrane around it. See, it engulfs it and forms this membrane around it. That's called phagocytosis. So essentially, phago means eating, right? And cytosis is cell, so it's cell eating. So phagocytosis is one example of endocytosis. It's one example of bringing in a pathogen into the cell. Okay. Now it does that by making these little arms and grabbing it. So we call that amoeboid movement. The action of making these little appendages, these little arms to grab it, is called amoeboid movement. Don't worry about this too much. It's just a word that was in the book. Now that it's engulfed it, so you can see here, it engulfed it, it did phagocytosis, now it's in this little vesicle. Now what it's going to do is try and kill it. The way it kills it is it fuses this little thing, this little, because right, it formed a membrane around it, it's going to fuse that with this little organelle called a lysosome. And remember, a lysosome is like the trash can of a cell. It's where you put everything you don't want. So it doesn't want this virus, so it's going to fuse it with this little lysosome. So it's going to, whatever's inside this lysosome is going to go into this, it's going to go and get into contact with this virus or this pathogen. And remember, the lysosome has a lot of dangerous things in it, a lot of things that are meant for destruction. Okay, it's meant to destroy anything in sight. So it fuses it. Now you can see this virus or this pathogen was just destroyed by the contents of the lysosome, right? And then afterwards, after it's destroyed, it will release it and I'll go on to its next target. So that is it. That's it for innate immunity. So in essence, somebody manages to get in, and whoever it is, whatever it is, because it's foreign, because it doesn't have the same uniform, it doesn't have the same antigen, it will be destroyed. And what will destroy it? A macrophage, which is a type of white blood cell. Macro, big, phage, eater. It's a big eater. It's going to eat whatever it sees. It doesn't care who it is. It's not specifically killing this one or specifically killing this one. It's killing whatever's getting in, and whatever pathogen. Okay, so that's it for innate immunity. So again, I'm gonna recap every time because it's a lot of information. I don't want to confuse anyone. So we got our pathogens. We got our first line of defense, right? We got our first line of defense. As long as that's intact, we're fine. If it's broken, our body's gonna try and do blood clotting. If something gets in before we manage to do blood clotting, we're gonna have our innate immunity, which is just this macrophage eating it as soon as it can. Okay, now what's next? What can there be more? Isn't that it? We just got rid of the we just got rid of our pathogen. Isn't that all to it? Almost. There's one more important thing. It's called adaptive immunity. So we just talked about innate immunity. So now we're going to talk about adaptive immunity. So innate immunity was non-specific. Remember the name I showed you here? It was non-specific. It didn't care about the pathogen. Whatever pathogen it was, it would get it. Okay? Now we are looking at adaptive immunity or specific immunity, also called acquired. So there's three names to this one. I don't know why they decided to do this to you guys, but you have to know three names, okay? I'll tell you the most common name used is like adaptive and innate immunity, but just because if you go onto a test and you don't recognize one of these names, you're going to be confused even though you already know it. So just for the sake of yourself, know these names. I know it's annoying, but that's just the way life is. Okay, so adaptive immunity. So this one is different. It's going to adapt, hence the name. It's not going to kill any pathogens it sees. It's going to be specific. It's only going to kill a specific one. Let me use this analogy to make this clear. So let's say there are many kinds of enemies outside these walls, all right? You can have one from one country. You can have one from another country, right? Let's say this war is fought by many countries, many teams. So this is from one team. He's from another team. He's from another team, whatever. You can see by their uniforms. This one is green, looks like he's about to throw up. This one looks cold, he's bluish. Okay, so anyways, they're all different teams. They have a different uniform. So what happens in adaptive immunity is this. Whatever person comes in, so obviously innate immunity can happen at the same time as adaptive immunity. So those macrophages will eat them or whatever, but adaptive immunity can also happen. And this one is basically looking at this enemy, analyzing it, and making a specific weapon to kill it as most efficiently as possible. So let's say this enemy comes in, this pathogen, and your team analyzes it and it sees, okay, this guy, his weakness is this and that, his strengths are this and that. Let's make this weapon because that will kill his army the best. So when his friends come in the future, his friends will be trained by the same army, by the same coach. So they will have the same strengths. So whatever weapon they just created to kill this guy, that weapon will be perfect for this guy. So they'll kill this guy even more efficiently. So basically, that's what adaptive immunity is. It is taking this pathogen, analyzing it, and making a weapon that is absolutely targeting its weakness and killing it in the most simple and sensitive way possible, instead of just attacking it by a standard way, okay? Instead of just attacking it by the macrophage. Because think about it. The innate immunity, it's just a macrophage eating these pathogens. That will take a long time if this macrophage had to eat one, go to the next one, eat it, go to the next one. That'll take ages. That's not very efficient. The adaptive and specific immune response analyzes this guy, goes and makes weapons, and then when they come in, they can just blast them and kill them instantly without killing one by one by one, right? It's more efficient, and you'll see what I mean. Okay. But when another person comes in from another army, that weapon that they just created, that might be useless against this guy, because this army, this person, this pathogen has a different strength, has a different weakness. So that's what's important about the adaptive immunity. It's very specific. It is very efficient at targeting one specific pathogen. It's very, very efficient at that. But when a different pathogen comes in, it may not work as well, okay? Now it has to repeat the same process and analyze this next pathogen, okay? But anyways, this is just the analogy. We'll look now at the real deal. Okay, here we are, the real deal. Take a deep breath. I gotta take a deep breath, man. Like, teaching all, like, I need to stay focused as well here. Okay, let's take a deep breath and then we do this one. Okay, this is a very, very key, very important one. So let's say we're now in our body, whether it's in our blood vessels, whether it's in our tissues surrounding our blood vessels, right? Because if we look at our, if we look at our blood vessels, there's also areas around your blood vessels that are empty, right? So these pathogens, not only can they go into your blood vessels, they can go into your blood vessels and escape from your blood vessels and go into the surrounding area. So, basically these macrophages, remember, let me go back to this. I want to emphasize this as well. Sometimes these pathogens can go out of the blood vessels and these macrophages can follow. So not all of the macrophages kill only in the blood vessels. A lot of the time, they actually kill outside of the blood vessels. They go outside of the blood vessels to kill, okay? Because that's where a lot of the pathogens go. Okay, so now, we're looking now at any place like the blood vessels, outside of the blood vessels, okay? We're looking at some area in the body, and our pathogen is there. So now I want to show you this adaptive immune response. So these cells here, okay, are called our B cells. The reason why they're called B cells, just for your information, you don't need to know this, is because they're made in your bone marrow, B for bone marrow, okay? Don't worry about that, you don't actually need to know that. Okay, and we have so many different types. I want you to understand this. So look at this B cell. It has this specific receptor. This receptor is meant for one kind of antigen, one kind of pathogen, okay? So let's say, and then this one has a different one, and this one has a different one. So we have so many different, we have such a wide variety of B cells, each one meant for a different target. So if we come to our analogy, each B cell has a different target. One will be specifically for this guy. One will be specifically for this guy. One will be specifically for this guy. So they're very specific. So if we look here, we have three beta cells here, and we have a pathogen. This one tries to recognize this pathogen. Oh, no, he doesn't. And I'm trying to do it by color. Notice this is like orangey yellow. So it tries to get it. I was like, oh, I don't recognize you. You're not my problem. The next one comes, oh, I don't recognize you. You're not my problem. This one recognizes it. So he's like, oh, I recognize you. You're my problem. So he's very specific because this molecule that he has is perfect for this enemy. Okay, he recognizes this enemy. So when he recognizes this enemy, two things happen. Two things happen. He wants to become, let me show you. He wants to, I need to make this clear. So he recognizes this thing, but before he can get activated, something else needs to happen. He needs another cell, okay? So he has this one kind of receptor. He has this one kind of receptor here that can hold this antigen. That can recognize another cell. So we have this T cell. So you got a B cell and you got a T cell. A B cell is made in the bone marrow, and a T cell is made in the thymus, T for thymus, okay, which is an organ in your body. Now, the B cell is specific to this pathogen, so it recognizes this specific pathogen, okay, but it's not going to kill it yet. It's not able to yet. It just recognizes it. So right now, it's like analyzing it. So it recognizes it and says, I got to take care of this one. But it cannot just start attacking it. It cannot just start being activated. Because what if it's not? It needs to be confirmed. So the T cell is going to come, and it also recognizes antigen. So here's this, you see it's the same antigen. So it picked up this antigen somewhere else in the body, okay? Because this pathogen can be everywhere. Because if you're never just infected with one, you're infected with many at once. So maybe this T cell went and found this pathogen. It picked up this antigen. So now what it's going to do is it's going to bring it to this B cell. It's going to bring it to this B cell, and it's going to show it to it. So look, it's showing it to the B cell. And it's going to say, oh, look, I also found this antigen. So I agree with you. I give you permission to go kill this thing. So it's basically the boss. It's like it finds it, and it goes to the B cell and says, okay, I give you permission to now get activated and destroy, okay? I give you permission. That's what the T cell does. So how does it give permission? It gives permission by sending little molecules to the B cell. Look, you see? It secretes these little circles. We call them cytokines. Cytokines. These are little molecules, little mail. It's like basically sending email. It's sending a bunch of words, little messages to this B cell, and the B cell will receive it and be like, oh, okay, so you agree, let me become active and kill this thing, okay? So very important, looking back at it. So the B cell recognizes this specific antigen, this specific pathogen, and it recognizes this antigen and says, oh, that's for me, this is my job, I'm made for this. But now, remember I said we have a wide variety of these different B cells, but of each B cell, we don't have that many. Very, very important. So even though we have so many different kinds of B cells, of this one, we only have like 5. We only have very few. That's not enough. That's very little. These 5 alone cannot sort out all of these pathogens that just entered your body. So when he recognizes it, it needs to be activated so that now many more can be made to destroy it. So the way it gets activated is it's first got to recognize this antigen on this pathogen, and the T cell needs to approve. The T cell will bring this antigen that it's found to prove that this one wasn't being delusional. It's bringing it and showing, okay, look, I found it too. You can get activated. And so it sends the cytokines. The cytokines will reach this beta cell and activate it. Okay, so now it will get activated. And what that means is this. Okay, it's going to get activated. What that means is it's going to start becoming very good at killing. Okay, it's going to, remember I said, it analyzes it because it wants to make a weapon. What is that weapon it's going to make? So it analyzed it because it wants to make a weapon so that it can make this weapon and destroy this thing as easy as possible. So to make that weapon, it needs to become activated. Activated means it's going to now duplicate its ribosomes. It's going to duplicate its rough endoplasmic reticulum. It's going to duplicate its Golgi apparatus. It's going to duplicate all these organelles and make them many so that it can remember, because all those organelles, if you watch the eukaryotic video, they are very important in making proteins. And this weapon that it wants to make is a protein. So when it gets activated, that's really what's happening. It's going to become slightly bigger because it's going to make a lot of these organelles so that it can make these proteins very efficiently. Okay, and not only, see, look at these little things that the little receptor that recognizes antigen. It's going to make a lot of these little proteins, these weapons, and it's going to release them to destroy these kind of pathogens. Okay, so not only does it become activated, it also starts cloning. So it's gonna duplicate a lot, because remember I said you only have a very few of these, and at this moment they're not activated yet. They don't make any proteins yet, so you can see it's empty. So once it gets activated by the confirmation from this T cell and recognizing the antigen, it will start being activated. So it's gonna now make a lot of these proteins, but not only that, it's gonna start cloning. So it's gonna duplicate a lot so that you can have a lot of these cells, and we call these cells plasma cells. And they have a very important job. They're the ones who's going to release these weapons so that these weapons can be everywhere in your body, and wherever these weapons find these pathogens, they'll kill them. Okay, so let me show you. So these plasma cells are going to pretty much explode. It's going to shoot out all these weapons. What do we call these weapons? We call them antibodies. Antibodies. Okay, so they are anti these pathogens. They're going to go target these pathogens and kill them by very different mechanisms. So an antibody is specific to an antigen. So this antibody is going to only kill this pathogen. It's not going to be able to kill any other pathogen. If another pathogen comes in, let's say it has a blue antigen, then this one would have to take care of it and make its own weapon, okay? So very important, these antibodies are specific to this antigen. It's only going to kill this pathogen. Very, very important. It's very specific. Okay, so how does it kill it? There's various ways. So if we say this is the pathogen, we're releasing these antibodies that's going to go all over our body, and wherever those pathogens are, it's going to demolish them. It can do one of three things. This antibody can attach to this pathogen's antigen, and by doing that, it's kind of like putting, you know how you can paste if you're being mean, you can paste a message on your friend's back as you hit him on the back, and it says "kick me" or whatever. It's kind of the same thing. These are little labels. They mark this pathogen, so it's easier for the macrophages to see them. So the macrophages, remember, they're able to eat them. So because it's now marking this specific pathogen, the macrophage can see it from far and say, ooh, that means I can eat it. So now it's going to eat it. Okay, very important. Another way is these antibodies can simply just destroy it. It can make holes in the pathogen so that they just die. Okay, and then lastly is they can form this weird kind of shape where they attach to the pathogens and form this structure, kind of making them all come together. So having a lot of pathogens in one small area. This is also efficient because now the macrophage doesn't have to eat one and swim all the way to another area in the body to eat another one. They're all now in one area. So this macrophage can eat one quickly. Okay, so that's efficient. So it's easier. So essentially this one makes it easier for phagocytosis. Easier for the macrophage to eat these things. And this one was for destroy. Okay. Now, another thing that can happen, so this one was so it can become this cell, which is the plasma cell, which makes the antibodies and helps to destroy this specific pathogen. It can also become what we call a memory cell. So this is a cell that lives very long and remembers very well. It has a very good memory. It will remember this pathogen very well. So it doesn't actually make anything. It's not like a plasma cell that makes antibodies and destroys it, but it has a very good memory. So it's going to remember this pathogen so that one day when you get infected again, it can wake up. And because it remembers this pathogen, it will now make these antibodies. It will then start working, which means that the second time you get infected, you will not get sick as much at all because you have these memory cells and they remember this pathogen from before. So they'll sort them out quickly because they remember them, okay? Because these plasma cells, they die pretty quickly. So they will make these antibodies and they will die. So next time it gets infected, they won't be able to do their job again because they won't remember. But these memory cells, they stay alive a long time. And the next time this guy comes in, these memory cells will kick in and take action. Okay, so that was a lot, I know, but I'm going to recap for you. So adaptive immunity or specific immunity is when you have a B cell, okay, not a macrophage or anything like that. It's a B cell, and it has this receptor that it's going to bind to the antigen. This receptor is specific for this kind of pathogen. It can only pick up this pathogen, and you have so many of these B cells, right? So you have thousands of them, thousands of these different B cells. And because there are so many pathogens in the world, you need so many different B cells so that hopefully one of them can recognize that pathogen, okay? Hopefully one of them. So in this case, this one recognized this pathogen. And now, because it is the one for that job, it needs a T cell to confirm it and give it permission. Once it's given permission, one of two things will happen. Some of them will become memory cells for the future when you get infected again. Some of them will become plasma cells, which is the one actually doing the work, making this very specialized weapon that's going to kill these pathogens very efficiently. And again, it is very useful because it can make millions of these very quickly. Instead of having one macrophage, right? Remember the innate immune system, one macrophage would eat one at a time. That's insanely slow. That's never going to work. So the adaptive immune system is adapting and making many of these antibodies so that it can sort out millions of these pathogens very quickly. Whereas in the innate immune system, a macrophage would eat one by one and it would only help a little bit. It would take, but in reality, it takes ages. So that's really the purpose of the adaptive immune response. Okay, so that's big. So let's recap once again. So we got what? What are these? Your pathogens, right? The pathogens, they're the hell those are the things causing you hell, including COVID-19, right? Which is a virus. We have our wall, our first line of defense. The wall needs to be penetrated before the pathogen can get into our bloodstream and cause us damage.
Okay, our skin can get damaged. Once that happens, we need to clot it up. We need to fix that wall so that we can prevent many bacteria from getting in. But sometimes bacteria do get in. What do we do then? We can do one of two things: innate immunity and adaptive immunity. And they kind of happen at the same time. They happen at the same time. So innate immunity will be our macrophages just eating whichever ones they see. No matter who they are, they'll recognize these antigens and just eat them, no matter who they are. But remember, it's a bit slow. It can only eat one at a time, move to the next one. It's slow, it's not efficient. It doesn't have weapons, it doesn't have bullets that it can shoot out, right? But then we have adaptive immunity, which is going to analyze. It's going to analyze this pathogen, the specific B cell, and once it's analyzed it, it's going to become a plasma cell, which is going to make this antibody, this weapon that's going to be shot out like bullets from a gun and kill all of these pathogens through different mechanisms, okay, or make it very easy for them to get killed. And they can also become memory cells. So that's our innate and adaptive immunity. Great, so that was a lot. So now we're going to look at a little graph. This graph is super interesting and very easy to understand if you understood what I just talked about. So it's kind of a way of summarizing what happens in our immune response. So here we have this axis, the x-axis is our time, right? And this is going to be the antibody concentration in our bloodstream. So kind of the amount of antibodies in our bloodstream. So let's say this happens. So let's say, okay, we get an infection. Some kind of pathogen managed to get into our body. Okay, this is our first infection, our very first infection. Okay, it gets into our body. Now what happens is, what? Our innate immune system and our adaptive immune system. So our adaptive immune system is going to do what? It's going to create all these antibodies to fight off this pathogen. So you can see here on this graph, after our infection, guess what happens to this line? It goes up. That means the antibody concentration is increasing, and that makes sense. So antibodies are being made. But look what happened. Look what happened. It's a bit slow. Look at this line. The slope is very slanted. It's not very straight up. It's quite slow. Over time, it's happening. The antibody concentration is increasing very slowly over time. So it's very long. So because it's so slow, there's a lot of time for your symptoms to develop. So the first time you get infected with something, you actually get sick. You have a runny nose, you have a fever, you have whatnot. That's because your body hasn't seen this pathogen before. So it's not very good at fighting it off. So the process of making these antibodies is a bit slow because look, it's got to recognize it, it's got to become activated and then make antibodies. So it's a bit slow. There's a whole process that needs to happen. It's to recognize it, get confirmation from the T cells, become plasma cells, make it, and then it can fight it off. So that's a bit slow, right? The first time it happens, it's a bit slow. So we call this our primary immune response. So it's the immune response that happened after the first infection. Okay, so let's say now this happened, our antibodies went up, we killed the pathogen, time passed, okay, we're healthy again, but now we get infected again by the same pathogen, the same one. It needs to be the same one, okay? So we get infected for the second time now, but this time, look, wow, we get our antibody production so fast and so much in such a short time. So we can say here that it's very short, this antibody production time. It took no time. And that means that the second time you get infected, there's no time for you to get symptoms. Your body will make all these antibodies so quick that it can fight off the infection before you even get sick. How is that possible? How is that possible, my friend? That is because of what? The magical memory cells. Remember, I just told you, in your adaptive immune system, you produce these memory cells as well. And these memory cells do what? They are there to remember. So the next time you get infected, they can recognize it like this. They will recognize this pathogen, and they are essentially already activated. They're ready. So they're essentially these cells, except they don't do anything. They just wait. They just remember. So now, the next time you get infected, you'll have a whole bunch of these memory cells. They will recognize this pathogen, and instead of needing to be activated by a T cell and becoming activated into this, they're already ready. They just have to find you, and then they can make these antibodies. So it's much, much quicker. So because of this, the second time is much, much better. So we call this our secondary immune response. And that's why when you get infected for the second time, you don't even know it. So a lot of the times you get infected and you get very sick. Most likely after that you got infected again, but you didn't even notice it because your body already had these memory cells. And these memory cells can be alive for months to years. So that keeps you healthy from this one kind of virus or pathogen for a very long time. Okay, so that's it. This is very important to understand this little curve. So the difference between these two peaks is the time, and the reason is because of these memory cells that developed from the first exposure. Okay, great. Vaccines, next topic. This one is very nice. It relates very nicely to what we just talked about. Okay, so a vaccine. Here we have a who, and we have a vaccine. So a lot of people don't like vaccines because of the needles. It's like, oh man, because of the needles. And a lot of people out there who are not educated don't even believe in vaccines. But I'm going to try and show you why vaccines are in fact important. Okay, so a vaccine is something you get, right, to prevent you from getting affected, to prevent you from getting serious consequences when you get infected by a certain organism. Okay, so let me show you. A vaccine, there's two ways a vaccine works. Traditionally, so it still works like some people still use this, some scientists. What is essentially in a vaccine? In a vaccine, you have a pathogen minus the disease-causing parts. So if we take this one here, if we say this is HIV, this is the human immunodeficiency virus. Okay? If we take that one, we don't want to put that in there and inject it into the person because that will just kill them because this virus is dangerous, right? We don't want to do that. We want to give it to the person without making them sick. How is that possible? Okay, remember, what is the thing that your body responds to? When you look at the adaptive immune response, this one here, what is the thing your B cells recognize? These antigens, right? Whatever the pathogen is, it doesn't look at the inside or anything, it looks at the outside. It recognizes these antigens. So essentially, doctors realized it is enough to just give an antigen. So what they do is they essentially they take this virus or this pathogen and remove an antigen from it. So let's say they took this thing and then they inject this into the body because this is not dangerous, but this will activate your body because these B cells will now recognize this antigen and make this weapon against it, just like I just explained. But now next time when the real HIV comes in, your body can fight it. Your body can fight it. When the real pathogen comes in, it already has all these memory cells, because this whole process happened, only it happened now without the real pathogen. Instead, it happened with just the antigen. So a virus is essentially a very nice way of just injecting an antigen, not the whole thing, just a part of the pathogen without it being dangerous. And it's injecting that into the person, and your body is going to think this is the real thing, and it's going to recognize it, do the whole adaptive immune response, making all the memory cells and all the antibodies, essentially as if it was the real thing. So now, next time, so if we look at this curve, so now you're basically, by getting a vaccine, you're getting this first dose, your body is making all the memory cells, all the antibodies, and sometimes you get a second dose and all that. So now, when you get infected with a real bacteria, the real thing, your body is already ready. You got all these antibodies, you got all these memory cells. There's no way you're going to get symptoms or get severely sick from it because your body is ready. So a vaccine, really, all it is, it's just a very nice, safe way. It's a way to give you the virus, to give you the pathogen in a safe way, okay? To give you a part of the pathogen instead of the real thing, the whole thing that makes you sick or can kill you. So that's the traditional way. And sometimes they like to call this the inactivated virus. Okay, so another way is advanced. So this is the more recent ways that they're doing. This is what they did with COVID. So what they do is, instead of giving an antigen, like the traditional way, and waiting for your body to react to this and make memory cells and antibodies to it, instead of doing that, they give the specific DNA or RNA that codes for this antigen. Because remember, this antigen, everything about a virus is made because of the DNA, because the DNA is the code. So instead what they do is, in the vaccine, they put the DNA and RNA for a particular antigen. So let me show you. So they inject the DNA or RNA that codes for a particular antigen. So your cells will take up this DNA and read it, right? Because your cells don't know if this is your DNA or someone else's DNA. They just take it up and they'll do what it says. Okay, it's like an instruction manual. So it will take up this DNA that's actually from a virus or a pathogen, and it's going to read it and do what it says. But scientists made sure that this DNA and RNA is coding for only one thing, and something that is not dangerous to the human body. So let's say this DNA or RNA coded for this antigen. So your body cells will make this antigen, and now that it's made this antigen, guess what? Your B cells will recognize it, again, make memory cells, make antibodies. Now, the next time you get infected with the real thing, now it comes into your body already has all the antibodies, has the memory cells, and so attacking this will be very easy to get rid of it. So that's all a vaccine really is. I hope that makes sense for you. So there's two ways that they generally work. COVID vaccine was some of the vaccines were RNA vaccines. So they work this way. They gave you a piece of RNA that your body cells read, and they made that antigen, the specific COVID antigen, not the whole virus, just a piece of it. And your body recognized that, made an immune response. So next time you really get infected by it, it won't be so bad. So that's it for vaccines. I hope that works. And you can see this curve is very, very similar to the normal immune response. So that's all that the vaccines are doing. They're causing this immune response in a safe way, because some viruses and some pathogens are very dangerous. We don't want them to attack us without already being ready, all right? Because if COVID attacks you from the beginning without you being ready, without you having memory cells, you can seriously suffer from it, because it's a very dangerous virus. So many pathogens are very dangerous, and we can't let our bodies deal with it by ourselves because we might die. Okay, so that's very important. That's why we care about vaccines. Okay. So up until this moment, we covered really how the immune system works very well. We talked about, I'm going to go to the first slide instead. I don't want to go through all those slides again for you. So we talked so far about pathogens. We talked about our first line of defense, how our skin serves as our first line of defense, and why this is important. We talked about what happens when our skin or our first line of defense is broken, and how our body fixes that by doing blood clotting. Sometimes our body cannot clot in time, and bacteria or any kind of pathogen can get in. In that case, our innate immunity and our adaptive immunity needs to kick in. And remember, our innate immunity is essentially our macrophages just eating all these pathogens randomly to get rid of them, whereas our adaptive immunity is mounting a specific, it's adapting, it's mounting a very specific response to this specific pathogen so that it can be an expert and make a very nice weapon or an antibody to eliminating that specific pathogen very efficiently. We talked about vaccines. So now we're going to soon talk about HIV and then do a bunch of IB questions. Okay, so if you're still here, man or woman, well done to you. We're almost there. Okay, so before we talk about HIV, there's one small thing here. It's called herd immunity. You know a herd. What's a herd? A herd is basically like a pack of animals, okay? A group, right? A group. So a group immunity essentially works like this. So let's say we have a country, right, and there's people in it, okay? Let's say this is a continent rather, it's Africa. We got some contagious people, meaning they're infected and they can spread the pathogen to other people. And we have these susceptible people, meaning they have never encountered this pathogen before. They have never been immunized. They have never seen this pathogen before, so they can get infected and suffer from it, right? So if you have a lot of contagious people and you have a lot of susceptible people, do you think all these susceptible people will get infected or not? Yes, they will. Because look, this guy can pass it to him, this guy can pass it to him, this guy can pass it to him. It's very, very easy for everyone to get infected. Okay, what if some people get immunized? So let's say we have some immunized people, these blue people. Can all these susceptible people become infected? Of course. This guy can pass it, this guy can pass it. It's very easy. So despite the fact that there's some immunized people, these other people can still get infected very easily, right? It doesn't help that only some people are immunized. But what if most people are immunized? Look at these blue ones. Most people are. Can these susceptible people get infected? Yes, they can, but very, very unlikely. Because look, this guy, let's say this guy, he cannot infect that guy because around him are so many immunized people that they won't get sick. So if they can't get sick, then how can that guy get sick? Because they can only get sick if they get in contact with these infected people, but they're not even infected, they're immunized. So this idea is that if enough people are vaccinated, then some people, even if there's people who are not vaccinated or not immunized, they are very unlikely to get infected because there's enough vaccinated people to keep them safe. Because these people that are contagious cannot reach them because even if, because this virus will not move from this person to that person and then that person, because this person can't get it. They're healthy. Do you see? So it's very hard for the virus or the pathogen to spread and kill susceptible people if many people are infected. So I'll put the words here. That's the idea of herd immunity. That's why countries really want most people to get vaccinated, because that's when the full effect happens. So that when poor people or people who don't want to get vaccinated or old people, they, even though they're not vaccinated, they're still relatively safe because of this idea of herd immunity. So I hope that makes sense. Okay, now real quick, we got to talk about, so we talk now about how your immune system, right, works. How it works when you're normal. So there's a disease called HIV that affects your body, and it actually damages your immune system. And when it damages your immune system, you're in big trouble, which means you can get infected too easily compared to a normal person. Let me show you. So HIV is called the human immunodeficiency virus. Why? Because it acts on humans and it makes you immunodeficient. That means your immune system becomes deficient, meaning not efficient, after you get infected by this virus. Let me show you how. So, let's say this is these HIV viruses, okay? The human immunodeficiency virus. What it does is it acts on T cells. Remember from our the adaptive immune system, the T cells were very important. Why? Let me show you. The T cells were very important to give permission to the B cells to start differentiating into memory cells and plasma cells so that they can make antibodies. So if your T cells are broken, what happens? Then your B cells cannot differentiate. They cannot become memory cells. They cannot become plasma cells, which means what? You cannot make antibodies, which means what? You're screwed. You cannot fight that infection. You cannot fight the infection that you're trying to fight. So essentially, HIV, this virus, is killing these T cells, which is causing this cascade of reactions, which means your immune system is compromised. You cannot fight infections so well anymore. So what happens is, over time, so initially you have a lot of these T cells, and because these HIV, these viruses, are starting to kill your T cells, gradually over time you'll have less and less and less T cells. When you have very little T cells, you call it AIDS. So AIDS is called acquired immunodeficiency syndrome. And it's called acquired because you get it. You get this virus and it starts causing this problem. You're not born with it, it's acquired. It's acquired immunodeficiency syndrome. So AIDS is caused by HIV. So these words are very commonly confused, HIV and AIDS. AIDS is the long-term consequence of being infected with HIV. Initially, you're still fine because you still have enough T cells. You can still fight things well enough. But after a long time, when your T cell level really drops, we call it AIDS. This is a point where your body is no longer coping anymore. So HIV is the virus, AIDS is the long-term consequence of being infected with HIV. Okay, and that means you reach a point where your T cells are so low, even they call them very weak pathogens. So a lot of pathogens in the world are weak. It's almost impossible for them to infect you. They're very weak, they're not strong, right? There's some pathogens that are very strong, some that are not so strong. So you get to a point where even weak pathogens can infect you, and we call that opportunistic infections. These things normally don't infect you, but now your immune system is so weak that even these ones can infect you, and we call that opportunistic infections. They infect you even though they're not normally supposed to. Oh yeah, so I just want to write here. So the cell that's being destroyed is a T cell, specifically a CD positive. Don't worry about that too much. Just know there's various types of T cells. The specific one is called CD positive T cell, but don't worry about that. Just know it's a T cell. So how do you get infected with an HIV? So HIV is interesting. You don't get infected the same way you do with COVID. With COVID, you have to inhale it from the air. You have to touch something, then touch your face and inhale it. It's not the same way. HIV works different. Look, HIV is transmitted. Look at my mouse here. There's a lot of shit going on here, a lot of stuff going on here. Let's look at my mouse. So when you look at your mouse, the mouse, look here. The way HIV is transmitted is through blood, semen, rectal, and vaginal fluids. Blood, semen, rectal, and vaginal fluids. So how does this work then? So the way you can get HIV is if someone who had HIV before, for example, someone who had HIV before used a needle, okay, from the doctor. Now the doctor didn't throw away that needle and use a new one. They take that same needle and they inject it into a new person. That way, you now transmitted that virus from the old person who had it into this new person. So that's one way. Another, because that's blood, right? It can be transmitted by blood. Another way is unprotected sex. So HIV lives well in your semen. So if you have unprotected sex, it can be passed on from one person to another. Pregnancy, that is kind of with blood, because the baby is connected to the mother with blood. So it can be passed on that way. And even with milk. So HIV can even be in breast milk, so the mother can pass it to the baby. So these are all ways in which HIV is transmitted from person to person. Okay, so what you need to know about HIV is what HIV does, the cell it affects, so it affects your T cells. By affecting your T cells, over time it causes AIDS. When you have AIDS, you get infected by every basic pathogen that you're not normally supposed to be infected by, and you cannot even fight them, so you die very easily. Very important. Okay, great. So that's it, lads. You did it. You did it. Now it's just questions. So if you can do these, I'm sure based on all this, you'll be able to do the questions. Let's just give it a try. So what stimulates the production of antibodies? So remember here, antigens are antibody generating substances, right? Because your body recognizes these antigens, and these antigens cause your B cells to generate antibodies. So they're antibody generating substances. So what stimulates production of antibodies? Not AIDS. AIDS is a long-term consequence of HIV. Antibiotics is not relevant. Anticodons is not relevant. It's antigens. So the answer is D. Antigens cause your body to make these antibodies to fight them, these weapons to fight them. Okay, what actions can phagocytes perform? So you can try them by yourself. So remember, a phagocyte is a cell that likes to eat, right? Phage is eating, so it's an eating cell. What's the eating cell that we learned about? The macrophage. The macrophage was part of the innate immune response, right? It would just eat everything. And it can also be a part of the adaptive immune response to eat the cells that are labeled with these antibodies. So what do? Do they make antibodies? No. Which cells do? Plasma cells, the B cells, they do. What about 2? They leave the bloodstream and enter infected tissues. I said that's true, right? Remember I said these macrophages, they can, when the pathogens exit the bloodstream into the surrounding tissues, the macrophages can also leave to catch them. Okay, so they can leave the bloodstream and enter infected tissues. That's true. They're allowed to do that. They can kill pathogens that they've engulfed. That's true. So 2 and 3 are true. So what does that mean? The answer is C. What is an effect of the HIV virus on the immune system? Okay, we just did this. Reduction in the number of phagocytes. No, they don't affect our macrophages, right? They don't. Reduction in the number of lymphocytes. Yes, that's true. They affect the T lymphocyte. They specifically affect or reduce the number of T lymphocytes. So it's this one. They lead to the increase in ability to form antibodies. No, they don't. They actually lead to the reduced ability, because by killing T lymphocytes are supposed to what? They're supposed to activate B lymphocytes, and B lymphocytes make antibodies. So in fact, the HIV causes a reduced ability to form antibodies. So they decrease in the ability to produce antigens. Nope, that's not even relevant. The answer is B. What is non-specific immunity to disease? So non-specific immunity is the innate immunity, right? That's the other name. So is it blocking prokaryotic metabolism of antibiotics? Nope, that's not what we talked about today. Production of antibodies by lymphocytes. No, that is adaptive immunity. That is specific immunity. Antibodies is specific immunity. That's what the B cell does. So it's not B. Endocytosis of pathogens by white blood cells. That's true. Remember, uptake, eating them, phagocytosis of white blood cells, specifically which white blood cell? The macrophage. So yeah, that's true. That's innate immunity. Production of cloned plasma cells. No, that's adaptive immunity once again. Okay, next. What is the sequence? So these are all past paper questions, by the way. I haven't said that yet. So what is the sequence of events in blood clotting after the platelets have been activated? Okay, so ABC. So let's see how good your memory is. Thrombin to prothrombin. Nope, not true. It's always prothrombin to thrombin first. So, okay, let's look at B. Prothrombin to thrombin, right? And then thrombin will convert fibrinogen to fibrin. So catalyze just means to speed up, to cause to happen. It means to cause to happen, basically. So prothrombin to thrombin, yes, and then thrombin will catalyze fibrinogen to fibrin. That's true. That's true. We looked at that here, right? Let's go back to this. Prothrombin to thrombin. Thrombin will catalyze fibrinogen to fibrin. Okay. Let's look at the rest. Fibrinogen to fibrin, which catalyzes thrombin to prothrombin. So that's basically the wrong way around. This section happens after this section. Okay, so fibrinogen to fibrin, which catalyzes prothrombin to, no, that's just completely wrong. So the answer is B. Okay, what is the role of activated B cells? So that could be plasma cells or memory cells. So they can secrete antibiotics in response to specific antigens. Oof, that's gonna trick a lot of people. They don't secrete antibiotics. Antibiotics are man-made, right, by doctors or scientists. Antibodies are made in your body. So this would have been true if this was antibodies. So it's wrong for that reason. It's not antibiotics. They activate T cells. No, they don't. T cells activate B cells. Release histamine. No, they don't. That's another kind of cell called a mast cell, but you guys don't learn about that in the IB. So it must be D. Multiply to form clones of plasma cells. Yes, and these plasma cells can make all these antibodies. Okay, I think this is the last question here. The graph shows, I tried to show a nice variety of questions so that you can see that all of the stuff I taught is relevant and will come up. Okay, so the graph shows changes in concentration. So this is of antibody in the blood of an individual subjected to 2 separate infections at time X and time Y. So this is the same curve we've been looking at, right? This curve, same curve we've been looking at. So what distinguishes the events occurring at Y from those occurring at X? The individual was infected with different pathogens at X and Y. No, remember I emphasized when we talk about this curve, the first one and the second one is the same pathogen, just a different time. This is the first time you got infected. This is the second time. It's not a different pathogen. So it's not A. Memory B cells are involved in antibody production at Y, but not at X. Yes, that's true. Remember, here is your primary immune response, where your body does not yet have memory cells. They do not yet have antibodies, so they're slowly starting to make those. So now, for the next response, you'll have these memory cells, which can react quickly and make a better response than the first time. So that's why that one is true. At point Y, we have these memory B cells, which we don't have at point X. So the answer is B. Let's just confirm the rest. There's no clonal selection involved at Y compared to X. No, they both have clonal selection. So they both will have these B cells becoming plasma cells. It's the same with both. There are no T lymphocytes involved at Y, but there is at X. No, that's not true. They both have T lymphocytes involved. Like they both have this whole process happening. This whole process, right? Both of them. The one just also has memory cells. So that's it. It's B. So that's it for this video. I really hope it was useful. As you can see, we covered all of these things, and yes, this is a big video and I expected it to be. That's because there's really a lot of things in this specific one, defense against infectious disease. These other 3 videos will be far shorter. They're about smaller topics, but I didn't want to include them into this video because I didn't feel they were that relevant to this topic. I wanted to make this its own topic, and these will be its own 3 videos, and they are all part of C3.2. So stay tuned for those. I hope it was useful. I hope this really helped you out, and I'll see you in the next one.